Cool. So I'm Richard, and I'm going to be talking about assessing evolutionary tempo across gnathostome evolution, as you can see up here. So I'm at Imperial College at the moment. This is based upon master's work that I did last year with Joe Keating and Phil Donoghue. So, as Marco has already told us, modern vertebrates are divided into two groups: the gnathostomes, or the jawed vertebrates, and the jawless cyclostomes. And of these two groups, the gnathostomes are way, way more diverse. They have far more different morphologies, whereas the cyclostomes are basically lampreys and hagfish, which are on one hand weird parasite things, and on the other hand weird slimy corpse-eating things. And that makes the gnathostome evolutionary transition of great interest to evolutionary biologists, as it chronicles this enormously successful group rising to kind of dominance of vertebrates. The problem being, as Marco has already explained, that we can't actually tell all that much about the morphological evolution of gnathostomes from only living taxa, because obviously there's not very much in between them. And to make this of further interest, there's also probably been some kind of genomic duplication event that happened along here. Genomic duplication events have been previously linked to bursts in evolution and attributed to the success of clades. Fortunately, the fossil record comes to the rescue and reveals a range of ostracoderms, or jawless, a paraphyletic grade of jawless fish that kind of chronicle the acquisition of characters on the gnathostomes stem. The placoderms Marco was talking about have been cruelly neglected in this diagram, but lie here between the osteostracans and this sharky guy there. So these appear to form some kind of paraphyly along the stem. Overall analyses have consistently found this, and this paraphyly appears to chronicle the acquisition of different things like bony tissue and paired fins and then jaws at the top that slowly kind of build up the gnathostome body plan bit by bit. However, despite the fact we know that this is probably the case, there hasn't really actually been an overview that tests the hypotheses of monophyly and things like that of these different groups. There's been group level analyses, but never anything that actually really took a proper view over everything. So the aim of this project was to assemble a data set that would allow us to do that. And also, with that, to test various hypotheses. So this, we made a supermatrix, basically, of these various different stem gnathostome groups. So we took these from various places and then combined them, all from the literature, and then combined into a big supermatrix that spanned the entire stem gnathostome, entirety of stem gnathostome evolution. So all of these jawless things are at a generic level for basically all of the well-known taxa, and then the gnathostomes are from a sort of representative gnathostome matrix comprising some placoderms, osteichthyons, acanthodians, and chondrichthyons. And this resulted in a data set with 254 taxa and 626 characters. So obviously, on making the supermatrix, we had to find a tree for it to be of any use. So that's the next step, finding this tree. So this was run in TNT, and it came up with a tree of 1,537 steps, which isn't in itself very interesting. But basically, we found that the supermatrix seems to confirm that you have this paraphyly of different things. Of particular interest are the fact that the anaspids, which are these groups that might be monophyletic or paraphyletic, are a paraphyly in this, you can see it there. And the thelodonts up here could feasibly be a paraphyly, but are a monophyletic group. This is a simplification of this big tree, obviously, because this is a bit complicated. We also, as well as using parsimony, used Bayesian methods over here, which found much the same. And also we reduced the taxa on the basis of completeness, which resulted in this matrix, which again has much the same result. One thing worth pointing out is, slightly weirdly, in the gnathostomes we get monophyletic placoderms, which is odd, and I think probably not right. But in the Bayesian analysis, we do get a paraphyly, so that's kind of up for grabs between these, to be honest. But other than that, everything was more or less the same between the Bayesian, and so we seem to have this quite clear-cut image of a series of groups forming a paraphyly along the gnathostome stem, chronicling their evolution. So now we've got this, what can we do with it? Well, we tested some hypotheses of topology on the tree. So this is possibly the most boring slide ever, but it basically shows results of topology tests. So we ran the tree as was, which resulted in a paraphyly, and also with constraints placed on various groups that could or could not be monophyletic. So, for example, we constrained the thelodonts to be monophyletic and paraphyletic, and found a shorter tree with monophyly, but only just. And neither the Templeton test nor the Kishino-Hashigawa test, which assess whether trees are actually kind of fundamentally different in topology, neither of them had a significant result. So it seems that while our tree does give us this view of a paraphily, it can't really actually rule either way on many hypotheses of monophily. Interestingly, it doesn't on the cyclostomes either, which has been a continuing contentious thing, whether or not cyclostomes are monophyletic. So what else can we do with it? We also ran it through disparity using non-metric multidimensional scaling, and this provides a different way, a kind of phonetic way, a different way of looking at the data set. And this is taxa plotted in terms of characters. So we've got the various jawless things along here, hagfish up there, lampreys there, tunicates and things over there, and then the gnathostomes all on their own over on this corner. So it seems that the data set is differentiating between gnathostomes and jawless things in the first axis of that and NDS, but in the second there's lots of variation in the jawless stuff as well. But what we were really interested in was looking at rate of evolution along this tree. Because the idea being that if we found a point along the tree where, with an increased rate of evolution, it might support the idea that this genome duplication led to a period of increased evolution which could be attributed to the success of the gnathostomes. So we used the methods of Lloyd et al. 2012, who did an evolutionary rate study on lungfish, Devonian lungfish, and using their code, we ran our thing through it. And so this is going to take a bit of explaining, but this is a branch likelihood tree. And so this is a tree from the previous analysis. Sorry, it's kind of split into two, so imagine that this bit feeds into here. It's a slightly dodgy shape for the screen. But then, so we took the tree, we dated it using earliest occurrence data from the fossil record for the various taxa in the tree. And then basically what this does is so the characters can be plotted onto the tree in different ways. And so you can accept two kind of opposite ends of that spectrum are Actran and Deltran character optimization. Actran assumes all the characters evolved as early as possible. Deltran assumes they all evolved as late as possible. And so these pie charts, the top half of each pie chart shows for the Actran tree, i.e. where the character evolution is accelerated, whereas whether the evolution is faster or slower than expected on that branch. So if it's green, it means it's faster than expected, and if it's red, it means it's slower than expected. And each half of the pie is for 1,000 runs, and obviously represents the proportion of runs that got each result, and the bottom half is Deltran, and the top half is Actran. And so when we did this branch likelihood test, what we'd be looking for if we expected a single genome duplication causing loads of morphological changes, one point on the tree, where you get loads of evolution on the gnathostome stem. We don't really find it. We find a period here at the base of osteostracans and jawed vertebrates where there does seem to be a fast region of evolution. Sorry, that's there actually. That's at the base of galeaspids, osteostracans, and jawed vertebrates. And then there's also a period in the gnathostomes, but then also there's faster periods at the base of the galeaspids there and the osteostracans there. So there doesn't really seem to be one point in the tree where all this evolution is happening. We also used another of Lloyd et al.'s methods, which was clade likelihood. And this, rather than testing whether the branch is faster than expected in terms of evolution, it tests whether the clade is faster than expected. So, for example, this node at the base of the jawed things is basically saying that all of this clade is evolving faster than expected. And for this one is all of this and all of these guys. So what we found was that basically all of the nodes, all of the successive clades in the gnathostome stem are faster than expected in comparison to jawless things. But there's not really a point at which one could say, here's a faster bit of evolution. There's another clever thing we can do with this, which is looking at character acquisition through time. So this is Actran and Deltran. Their character acquisition rates plotted through time, and this is cumulative character acquisition, this thing here. And these are time-binned character, like bins with character acquisition in. So you can see most of the characters are gained in the Cambrian, which is not surprising because that's basically where our tree starts. But there's also this period in the Ordovician/Silurian where we have a high period of character acquisition. And that's probably attributable to the fact that most of the taxa, most of the groups we're working with originated there, so all the osteostracan and galeaspid characters and things. I suspect this is probably actually giving it later than it actually would be because the fossil record of stem gnathostomes is notoriously kind of dodgy in the Ordovician. We only really have some groups' data. So that's probably a slight estimate too soon. Okay. So, conclusions. What can we conclude from this? Stem gnathostomes, as previously found, resolve into a series of monophyletic and, in the case of the anaspids, paraphyletic groups, but they do sit along the gnathostome stem in a fairly uncontroversial manner, as has been previously suggested from group level analysis. Our data set isn't decisive on questions of monophyly, which is not ideal in terms of many of the stem gnathostome groups, like Thelodonts. It'd be nice to know if they were monophyletic or not, because it's going to continue to be a source of argument. But for things like the cyclostomes, where molecular data has been telling us that they are monophyletic for ages, whereas the morphological data continues to tell us they're not, it's interesting to see that from a morphological data set, it can't actually rule either way on their monophyly, even if it finds them as a paraphyly. Evolutionary rates don't appear to show one area of increased character acquisition that might correspond to a genome duplication causing loads of character acquisition, at least not as far as I can tell from our tree. And their characters seem largely to have been gained during the Cambrian and Silurian. That's pretty uncontroversial, but it's a nifty thing we can do with our time tree. And so we've got some future work planned with this. We're currently replacing the gnathostome part of the data set with a recent gnathostome analysis that came out, which has better character coding, and that ought to hopefully sort the gnathostomes out to some extent, because currently I think the weird placoderm thing is making it a bit dodgy. And then we're also planning on integrating it into a total evidence morphological molecular analysis, which ought to provide a tip-dated tree that spans the entirety of vertebrate evolution. This is actually quite a boring picture, but this is the molecular tree that we have for our various living taxa that we're going to put into that. So we're going to merge those two and hopefully get some idea of the timescale on this kind of thing on something other than fossils. Well, including fossils, but also using molecular evidence. Okay, so thank you to Graham Lloyd, Bradley, and Henning Blom for helping me with methods and data sets and things, and to Emma Bernard and Stig Walsh at various museums, the NHM and the MMS, for access to specimens. And I'll leave you with this picture of a Ordovician seascape. That's it. Questions?